Compound lipids, compound lipids. Compound lipids are the part of lipids, and these compound lipids are present mainly in the plasma membrane as a functional role. They are performing many functions and structural stabilities of the plasma membrane. Compound lipids are part of lipids, and these compound lipids are usually present in membranes. Membranes are plasma membrane. Plasma membrane. Many of the compound lipids are structural lipids of the membrane. Many of the compound lipids, or almost all the compound lipids, are structural lipids of the membrane. Structural lipids of the membrane. These compound lipids are divided into 5 types. 1. Glycerophospholipids. Phospholipids. Second, glycolipids. Third one, archaeal tetra ether lipids. Archaeal tetraether lipids. Fourth one, sphingolipids. Fifth one, sterols. These are the main compound lipids present in the membranes, or as present in the membranes as structural lipids of the membrane. These are 1. Glycerophospholipids, glycolipids, archaeal tetraether lipids, sphingolipids, sterols. We'll see one by one. Glycerophospholipids, glycolipids, archaeal tetraether lipids, sphingolipids, sterols are the structural lipids of the membrane, or membrane structural lipids, are also called as membrane structural lipids. Membrane structural lipids. In glycolipids, again two types: galactolipids, galactolipids, and sulfolipids. Together called as glycolipids. We will see glycerophospholipids. The first one is glycerophospholipids. Glycerophospholipids have three components. Glycerophospholipids have three components. One, glycerol. Second one, fatty acid. Third one, phosphate moiety. Glycerophospholipids have three components. One, components in the glycerophospholipids. They are also called as phosphoglycerolipids. Glycerophospholipids are also called as phosphoglycerolipids. These phosphoglycerolipids have three components in the one, glycerol as backbone, fatty acid as hydrophobic polar molecule, hydrophobic non-polar, phosphate hydrophilic. Phospholipids are glycerophospholipids. Glycerophospholipids. Glycerophospholipids have three components. One, glycerol, CH2, OH, CH, OH, CH2, OH, glycerol. Second one, fatty acid, it may be palmitic acid, or it may be oleic acid. This palmitic acid is saturated aliphatic saturated fatty acid of 16 the carbon number present in the aliphatic chain, and 0 is the no double bonds present in the aliphatic chain. Carbon number. And oleic acid is unsaturated fatty acid of 18 carbon and 1 unsaturated at delta 9. At ninth position, it has one double bond. And third one is phosphate moiety. Phosphate moiety. Glycerophospholipids have three components. One is glycerol. This is glycerol. Second one is fatty acid. This fatty acid may be saturated or unsaturated. For example, saturated palmitic acid 16,0 and unsaturated fatty acid 18,1 and one phosphate moiety. These three together are called glycerophospholipids. In the glycerophospholipids, the glycerol is the backbone of the lipid. Backbone of the lipid. Phosphate is the third component in the glycerophospholipids. Phosphate is the hydrophilic polar molecule. This fatty acid is the hydrophobic fatty acid, may be saturated or unsaturated. Saturated fatty acid or unsaturated fatty acid is hydrophobic. Non-polar molecule. This is glycerophospholipids. Glycerophospholipids have three components. One, glycerol has backbone. Second, fatty acid as hydrophobic non-polar. Third one is phosphate, is a hydrophilic polar molecule. This all together one glycerol, two fatty acid, three phosphate all together will form glycerophospholipids. These glycerophospholipids are amphipathic. Amphipathic in nature. As in amphipathic, we already know as the hydrophilic hydrophilic polar molecule on outer side of the plasma membrane, outer side of the plasma membrane. This is a typical plasma membrane structure. This is hydrophilic polar hydrogens. This is hydrophilic polar hydrogen contain phosphate moiety. Polar hydrogen contain phosphate moiety. This is also hydrophilic polar hydrogen. Hydrophilic polar hydrogen. This is the middle one is hydrophobic tail region or fatty acid component. Hydrophobic phobic tail or fatty acid component. This dual nature of the plasma membrane or phosphoglycerides make them as amphipathic in nature. Amphipathic in nature. We'll see the structures of glycerophospholipids. Depending upon the number of molecules varies, the phospholipids have different types. In phospholipids, this is in phospholipids, the fatty acid is attached to the glycerol through ester bond. R1, R2. Fatty acid chain 1, chain 1, fatty acid chain 2. This is phosphate moiety. Phosphate moiety. This is phosphatidic acid. The X may be in X place. If we put H hydrogen, it is called phosphatidic acid. Glycerophosphatidic acid. We'll see how this phosphatidic acid or glycerophosphatidic acid, fatty acids are in attached. We'll see. One, two, three, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. No, we'll write small. We'll write in small. 1, 2, 3, 4, 5, 6, 7, 8, 9, double 1 at 9th position, 10, 11, 12, 13, 14, 15, 16, 17, 18, 18. This is oleic acid. Oleic acid is unsaturated fatty acid of delta 9 unsaturated fatty acid This is palmitic acid. One, two, three, four, five, six, seven, 8, 9, 10, 11, 12, 13, 14, 15, 16, 16. This is phosphatidic acid, glycerophosphatidic acid. Glycerophosphatidic acid. In the glycerophosphatidic acid, if we replace the hydrogen with ethanolamine, ethanolamine ethanolamine, then it is called glycerophospholipid ethanolamine. Then it is called glycerophospholipid ethanolamine. If we place in hydrogen place of phosphatidic acid, if you place ethanolamine, this is the structure of ethanolamine, then it is called glycerophosphorethanolamine, or simply phosphoethanolamine. Glycerophosphoethanolamine. If we put in place of hydrogen, if we put choline, this is choline, glycerophosphoethanolamine, choline. This is choline. This is phosphodiester bond. Ester bond. bond or phosphoester bond. This is also called ester bond. Ester bond. This is also called ester bond. Ester bond. If we replace this is glycerophosphatidyl in place of H. We usually, if choline is not there, here hydrogen will be there. If we replace hydrogen by the choline, then the phospholipid or glycerophospholipid is called as glycerophosphatidylcholine. Earlier, phosphatidylethanolamin, glycerophosphatidyl ethanolamine and this glycerophosphatidylcholine and glycerophosphatidylserine serine glycerol and mono glycerophosphatidyl ionocetal phosphatidyl glycerol. All these compounds are compounds of glycerophospholipids and these are present in structural lipids or membrane structures. We'll see this is glycerophosphatidyl choline. This is glycerophosphatidyl choline. If we replace with choline, if this is phosphatidic acid, this is phosphatidic acid. If we replace the hydrogen with serine, we are replacing this hydrogen of phosphatidic acid with serine. We're replacing serine. Serine, a amino acid. Serine is replacing this hydroxyl hydrogen bond, and this will form phosphatidylserine. Phosphatidyl glycerophosphatidylserine. This is glycerophosphatidylserine. This is called glycerophosphatidylserine or phosphatidylserine. Phosphatidylserine. Next, this is hydrogen of phosphatidic acid. This is hydrogen of phosphatidic acid. If phosphatidic acid can also be replaced by glycerol. This glycerol can also be replaced this hydrogen. If this hydrogen is removed, and H2O, HHO, H2O as water. This is water of elimination. Then can have we can have phosphatidyl glycerol. For all the remaining like phosphatidyl, phosphatidic acid, acid, glycerophosphatidic acid, glycerophosphatidylserine, serine, glycerophosphatidylserine. phosphoryl ethanolamine, glycerophosphoryl choline, for all these phospholipids or glycerophospholipids, the aliphatic chain, the aliphatic chains, this is aliphatic chain or fatty acid chain may be variable. Depending on the aliphatic chain variable changes, the fatty acid chain is the phospholipid name and its function will be different. This is one important point. This is glycerophosphoryl glycerol. Glycerophosphoryl glycerol. Glycerol. And this is phosphatidic acid as H is there, and this phosphatidic acid is also be replaced by inositol. 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 In place of H, this inositol can also be accommodated, and then it is called through phosphodiester bond. Then it is called glycerophosphoryl inositol. Glycerophosphoryl inositol. This is glycerophosphoryl inositol. This all the components are called glycerophosphoryl inositol. And again to this phosphoric acid, one more phosphatidic acid can also be accommodated, then that is called diphospho phosphatidic acid, diglycerophosphatidic acid, diglycerophosphatidic acid, phosphatidic acid. Acid or glycerol. Then this is called cardiolipin. Lipid, cardiolipid. This is phospholipids or glycerophospholipids, different types. We'll see some important points regarding glycerophospholipids. Glycerophospholipids are having two fatty acid chains, two fatty acid chains, two fatty acid chains through ester bonds. Ester bonds. In some animals, and unicellular organisms. One ether bond will also be present along with ester bond. One ether bond will also be present. In vertebrate, heart tissue is enriched with the vertebrate heart tissue is enriched with these glycerophospholipids through ester bonds. And also through ether bonds. Membranes of halophilic bacteria ciliated protist ciliated protists have high proportion of ether bonds. Ether bonds. In the presence of these ether bonds is not yet to be known. The presence of ether bonds in glycerophospholipids is not yet to be known. But one of the glycerophospholipids have have one of the glycerophospholipids have platelet activating factor PAF is released by leukocytes sites of basophil cells is one of the activating signaling factor and this signaling factor will release serotonin, a vasoconstrictor is in heart tissue, smooth muscle cells, lungs, heart cells, liver cells, urinary or cells, urinary cells is this platelet PAF factor is controlled. And this PAF factor is also be leads to inflammation in the body. Phospholipids characteristics: the presence of ether bonds along with ester bonds. Some phospholipids have ether bonds also. Ester bonds. Some phospholipids will activate secret platelet activate these ether bonds. The presence of these ether bonds is not yet to be known. Some glycerophospholipids in membrane structure will activate platelet activating factor. This is a signaling factor. This signaling factor will release a vasoconstrictor serotonin. This is released by leukocytes, particularly basophil cells. Released vasoconstrictors. This vasoconstrictor is uh, imposed on smooth muscle cells, lung cells, heart cells, liver cells, and urinary cells also. Along with these imposing of these cells, this PAF factor will also be responsible for inflammation in the body. We'll see in the next.